In this video, I'll be going over a summary of the major events that happen in contraction. So shown here is a neuromuscular junction with this neuron coming down and branching in its telodendria and synapsing with the muscle cells. Each one of these is a muscle fiber or myofiber or muscle cell. The neuromuscular junction is shown here. We have a motor end plate, which is an indentation of the sarcolemma, which is the muscle cell membrane. And then you have the synaptic end bulbs shown here. They're going to contain acetylcholine. Now we're going to look at the events that happen at the neuromuscular junction. Action potential is coming down this lower motor neuron. This is the axon of that lower motor neuron. And when it gets to the end, at the synaptic end bulb, also called the terminal bouton, it's going to stimulate the opening of voltage-gated calcium channels. Calcium is going to come in, and it's going to stimulate the release of acetylcholine-containing vesicles by exocytosis. The acetylcholine is going to go across the synaptic cleft and bind to the acetylcholine receptors, which are also called ligand-gated sodium channels, because when this acetylcholine opens these receptors, sodium comes in. It comes down its electrochemical gradient into the muscle cell, where it's going to create a graded depolarization. Now, normally the muscle cells of skeletal muscle have a resting membrane potential of about minus 85 millivolts. As that sodium comes in and creates this greater depolarization, it can raise that minus 85 millivolts to threshold or minus 60 millivolts for voltage-gated sodium channels. Now, voltage-gated sodium channels are right along here on the sarcolemma. If those reach threshold, then they're going to open, and you're going to generate an action potential down the muscle cell membrane. Well, before we go on with the action potential down the muscle cell membrane, let's take a look at some more of the structures here at the motor end plate. The motor end plate is the collection of acetylcholine receptors on the muscle cell membrane where the axon terminal, makes a synapse with the muscle cell. So at this motor end plate, you're going to see a lot of folds. These are called junctional folds and help to increase the surface area. You get a lot more acetylcholine receptors bound here. And then around those acetylcholine receptors, you also have an enzyme called acetylcholine esterase that helps break down the acetylcholine once it comes across and binds to the receptor. When you form that action potential down the sarcolemma, it's going to move down sarcolemma, open up voltage-gated sodium-potassium channels, and then it will continue down through these T-tubules. Now notice the T-tubule comes in and goes right along or next to that Z-disc of the sarcomeres. Now on each side of the T-tubule, we're going to see that there's a sarcoplasmic reticulum. So here is a T-tubule. Here's a sarcoplasmic reticulum. That's going to be a storehouse for calcium. Well, the part of the sarcoplasmic reticulum that's right along the T-tubule is called the terminal cisternae. It's an expanded out portion of the sarcoplasmic reticulum. And in that terminal cisternae, the membrane, you're going to have these calcium release channels that are physically connected to the voltage-gated dihydropyridine receptors. And those are here in purple. And they're part of the T-tubule membrane. So when that action potential comes down that T-tubule, it's going to change the voltage across the T-tubule due to the opening of sodium channels, which is going to stimulate this voltage-gated dihydropyridine receptor to change shape, and that's going to physically open the calcium release channel. Now, calcium release channels are also called ryanodine channels. So on the next slide, here it shows the DHP receptor that's physically connected, this is part of the DHP receptor, to this calcium release channel. Now in this particular image, you will see that there's quite a bit of a space between this calcium release channel and the T-tubule. Now that's normally not the way it is. These are physically connected and there's not a lot of space between them. So for example, here's your T-tubule, here's the T-tubule, and the sarcoplasmic reticulum on each side. Well, remember, the expanded out area of the sarcoplasmic reticulum is called the terminal cisternae. So when you have a T-tubule flanked on each side by the terminal cisternae, we call this three-bodied structure a triad. And in the membrane of the T-tubule, you'll see these dihydropyridine receptors shown here. The calcium release channels shown here are part of the membrane of the terminal cisternae, just inside of this terminal cisternae, or sarcoplasmic reticulum. Now, calcium release channels and the DHP receptors are physically connected and they're extremely close together. Like I said, when that action potential comes down, that T2 simulates a dihydropyridine receptor to change shape. It's going to pull open the calcium release channel, and calcium that's stored in the sarcoplasmic reticulum is going to come out into the sarcoplasm, and then it's going to bind to troponin, specifically troponin C of the troponin tropomyosin complex. Well, this troponin tropomyosin complex is covering over the active sites for the myosin head. When calcium binds, it's going to cause a conformational change or change in shape of the troponin molecule, which is then going to pull the tropomyosin out of the way and expose the active site or attachment site. The myosin head, which is cocked back and ready to go, we're going to see ATP previously cocked it back, and when you have a relaxed muscle, those myosin heads are cocked back and waiting for the active site to become available. As soon as calcium binds to the troponin C, changes shape, moves the troponin tropomyosin complex out of the way, exposes the active site, the myosin head is going to bind and power stroke, moving the thin filament over the thick filament. ATP is going to come in and bind to the head, break the bond between the thin filament and the myosin head, and cock the head back. When it does this, as long as calcium is still available and the attachment sites or active sites are open, the myosin head will continue to bind and power stroke, as long as ATP is there to cock that head back. So shown here is the troponin. This is a tropomyosin in blue, and this troponin that's in pink has three parts. It has a troponin T, the T is for tropomyosin. That's the part that attaches the troponin to the tropomyosin. You have troponin C, C for calcium. This is where calcium is going to bind to the troponin. You also have troponin I, or inhibitory, and this portion can cover over the active site and block the myosin hair from binding. However, most of the blocking occurs with the tropomyosin. As you can see, the calcium comes in, binds to the troponin C, causes a shape change in troponin, which results in the troponin tropomyosin complex to be moved out of the way of the active site. Here we have the myosin head. The myosin head is going to come in, it's going to bind and power stroke. This green ball right here that's coming in is ATP. When ATP binds, it releases the energy into that myosin head that causes it to cock back. Well, once it does this, it's going to be changed from ATP to ADP and phosphate. And that phosphate's going to come off, and the ADP will come off. So let's take a look at that. So here's a phosphate coming off. And then here's the ADP coming off. Well, notice the myosin head is already cocking back. Notice the myosin head is already doing a power stroke. And what's going to release the myosin head is this new ATP coming in to bind to the myosin head to cock it back. Right there. Now, as long as calcium is there and the active sites are available, it will continue to do this over and over and over again. So if we look at this, these are the myosin heads here, shown here and here. These are the thin filaments. Here's a thin filament. Here's a Z disc, another Z disc, just as a diagram here. Well, as these myosin heads power stroke, they're going to pull the thin filaments over the thick filaments, causing the sarcomeres to shorten. So let's take a look at this animation of a sarcomere contraction, which will give you a good visual on how all the sarcomeres shorten during a muscle contraction.
In a relaxed muscle, actin and myosin myofilaments lie side by side, and the H zones and I band are at maximum width. During contraction, the actin and myosin myofilaments interact. The actins are pulled toward the center of each myosin myofilament. As a result, the sarcomeres shorten. In the fully contracted muscle, the ends of the actin myofilaments overlap. The H zones disappear, and the I band becomes very narrow.
So what causes a muscle cell to relax? Well, when you stop sending action potentials down this neuron, you're going to close the voltage-gated calcium channels, which is then going to cause the acetylcholine-containing vesicles not to be released. With the acetylcholine here that's bound, it releases, and then you have acetylcholine esterase that's going to break down the acetylcholine that's here. You're no longer going to open up the acetylcholine receptors, no sodium goes in, so you don't have a greater depolarization here, which means these voltage-gated sodium channels stop opening. The active potential ceases to go down the sarcolemma, it ceases to go down through the T-tubule, and then the change in voltage back to the resting membrane potential causes these dihydropyridine receptors to change shape back to their normal shape, which is then going to close a calcium release channel. Once a calcium release channel is closed, the sarcoplasmic reticulum is going to use these calcium ATPase pumps that are all along the sarcoplasmic reticulum, and they're pumping the calcium back in from the sarcoplasm into the sarcoplasmic reticulum. This is against a concentration gradient, so you have to use energy in the form of ATP to pump this calcium back, hence the name a calcium ATPase pump. Once the calcium comes off the troponin, that troponin-tropomyosin complex will cover back over the active site. The myosin heads cock back, but since the active site is now covered up, they're cocked back, ready to go, and there's no more binding to the thin filament, so the muscle relaxes. If you have any questions of this, don't hesitate to ask.